Melon, please. Okay, thank you. Yeah. Okay, so the previous talk, Peter already talked a lot about why microRNA is important, so I'm just going to briefly going over this one. So we're working on a problem of trying to identify gene targets of microRNAs, and this is an important step to understand microRNA regulation. So we know that microRNAs is a both transcriptional gene regulators, and it has been associated with a growing list of human disease. So it's important potential biomarkers and therapeutic targets. And also more than half of human genes are targeted by microRNA, so make them very important. So there's a lot of computational approaches to these kind of problems. The first set of approach, people try to find sequence-based features. So to look for discriminate sites of microRNA binding. For example, look for heavy structures, looking for conserved features among mutable species. However, this set of approach have a very high false positive rate. The main reason because microRNAs are tissue and condition specific. Famous example here is that the microRNA 124A is restricted to the brain and spinal cord in both zebrafish and mouse. However, microRNA 1 is active in the muscle tissues of zebrafish and the heart in mouse. So we need more than just sequence-based or static features to be able to capture this type of dynamics. So the second set of approach proposed to tackle this problem. So they try to integrate sequence features with expression data. So we can think of this as a regression problem. So here we have, why is the expression values of the mRNA that we try to infer what microRNA targets it. And we have a bunch of microRNA X1, X2, up to Xn, and we try to express or explain the expression values of Y in terms of X1 up to Xn. And the target prediction problem becomes the model selection problems where we try to find which X is a potential or give us a prediction power to regress Y. So people look at many type of relationships, for example, Pearson, Spearman correlation, mutual informations. Also people use linear regression, lasso regression, completely linear Bayesian models to this problem. Okay. So far we have been looked at each microRNA individually. So each of them is a separate regression problem. So we all know that microRNAs cooperatively or coordinatedly target protein complexes. And there's a lot of evidence suggesting that interacting proteins are co-regulated by the same microRNAs. So we want to propose work that extend the expression and sequence data with interaction data. In ISMB 2001, there's one method that's based on non-negative matrix factorization. It's try to find the modules of microRNA and mRNAs. In that modules, the mRNAs are close in the PPI or protein-protein interaction networks. Okay. So we extend this CAP idea further, and we propose this method called protein interaction-based microRNA modules, short as PIMIM. So in here we have three different types of data. So here we have X and Y as expression data of microRNA and mRNAs, and phi is the list of potential interactions between microRNA and mRNA. So we can get this list from a lot of available database. The third one is we use protein interactions database. So it's also a list of pair proteins. There's a lot of database that we can take advantage here. So the output of our model is a modular network of microRNA and mRNAs. So each module would correspond to a set of microRNA and mRNAs that have some regulatory relationship. Okay, so I'm going to go to more a bit technical details here. So we use this very simple matrix representation. So the module membership would be represented by two binary matrix, U and V. So U is for microRNA and V is for mRNA. And each column corresponds to one module, one feature. So at the end, if we look at module K, it would be the kth column of U and V. And we restrict them to be non-negative matrices. Just, so that for easier interpretation later in the model. Okay, so how we incorporate the data into our model through the likelihood. So we look at each microRNA Xi and mRNA Yj. We can think of the row UI and Vj as a module membership of microRNA Xi and mRNA Yj. And then by taking the dot product of UI and Vj, this can be thought of as the interaction strand between Xi and Vj. Notice that this dot product would be 0 if they have no shared membership, so they don't belong to any of the modules together. And this could be of large value if they have many modules in common. Okay. So using this, we can incorporate the likelihood. So there's two parts. First is the expression data. So the expression data, probability of Y given U, V, and X would be the product of all the mRNAs, J. And each expression of mRNA would be expressed in terms of using a normal distribution. And we use the expression values of all the microRNA Xi. And then we use the interaction strand that I just described previously to tell how strongly the microRNA would, the microRNA would be affecting the mRNAs. Okay. The second part is how we incorporate the interaction data we have from the two matrices, Phi and omega. So Phi, if we think that the microRNA Xi would interact with the mRNA Yj, then we want to encourage them to have the same modules, encourage them to belong to the same modules. So we use a logistic function to do that. If we don't think that, if we don't have a prior that they should interact, then we just 1 minus the logistic function to saying that. Similarly, if we think that two mRNAs, Yj or Yj primes, interacting in the PPI networks, then we also use the logistic function to help us to encourage them to be in the same module. Okay. Together, all this, we pose this optimization problem. We try to minimize the negative log likelihood, and we add 2 extra constraints for sparsity, such as the L1 constraint. Because this, we restrict that U and V are non-negative matrix, so projection of U and V on a non-negative L1 number is easy, so we can solve these non-convex problems using a projected quasi-Newton method. And we need to do randomization with different starting points to get over the non-complexity. Okay, so this is evaluation. So we use a data set of 385 samples of cancer patients. So first the first question we ask is, do the microRNAs that we think are active in this data set are meaningful, right? So we use a data set of 115 cancer microRNAs that we know that those microRNAs were determined to participate in ovarian cancer. And we look at the precision recall values of, so precision is how many truths positive over the total numbers of microRNAs that we inferred, and recall is how many the true positive divided by the total, divided by 115 in this case. So in all of the counts, F1, precision, and recall score, we outperform this non-negative matrix method I mentioned previously. Okay, the second question we ask is, does the modules that we get represent something or have any meaning to it? So we use a set of 880 gene set from MSIC database. So this is a curated set of genes that they known to participate in different pathways. So we use multiple hypothesis testing, correction for multiple testing, and vary across different false discovery rate cutoff on the x-axis. And so the top plot shows the number of modules that are enriched by at least one of those gene sets. And the bottom plot shows how many of those gene sets are enriched by at least one of our modules. Okay, so in both of those two plots, our method is doing better than the non-negative matrix factorization method. Okay, so far, I've been trying to infer one set of modules for one condition, or in our case, in the previous result is one specific type of cancer. We want to extend this model further to do a joint analysis of multiple types of cancer. First of all, what we want to find regulatory modules that are differently, they work differently in different type cancers, or we can find modules that work similarly across different type cancers. So we want to do that. So here, instead of learning one set of modules, we learn T set of modules for T conditions. So how we do that? So for example, here we have 3 conditions. So we want to learn U1, U2, and U3. So the notation is that U star IK corresponds to the IK entries of the matrix U1 up to UT. And similarly, V star GK is the GK entries of the matrix V1 up to VT. So we can add an additional regularization terms to encourage that the elements of U star VIK and V star JK to be selected together. So this is an instance of multitask learning. And by doing this, we can compare the set of modules across different conditions. Okay, so let's see if that's, sorry. So then this is some early results that we get using this model. So we take 3 cancer types, breast cancer, glioblastoma, and leukemia from TCGA websites, and we do joint analysis of those samples. The MicroRNA and mRNA interactions was downloaded from the 3 public databases, and the protein interaction is downloaded from Transfact. Okay. So this is the module memberships of the MicroRNA. So MicroRNAs are shown on the y-axis, and the x-axis shows the modules. So each color corresponds to one type cancer. So you can see that we can identify modules that are very similar, have very similar membership across 3 cancer types, and we also identify, for example, this one module that only activate in 1 or 2 type cancers. So this is kind of the power of the method that we can identify. Okay. In particular, I'm gonna talk in more detail about these modules that we found. Okay, so this is the module that we found. So we call this one cell proliferation module. So the color here encodes wherever the interactions was found in all 3 types of cancer, or it was in 1 or 2 type cancer. So the thing in the middle is the genes, and the outer circle are microRNAs. So we found that all the microRNAs that we found here in this modules are all reported previously that related to cancer. For example, famous like 200 ABC cluster, 141, 429 are all like related to cancer and cell proliferation. Interestingly, they are located in 2 regions that would support our prediction that they cooperatively regulate those genes. If we look at those genes, we can find there's one famous tumor suppressor genes, like the breast cancer genes. And this set of genes correspond to the TFTC and TFFID complexes, also been reported previously that related to cancer. Okay, so just short. In summary, so we develop a method that inferring condition-specific regulation of microRNAs for identifying targets. So this method combines sequence expression and interaction data to discover microRNA regulated modules of mRNAs. And we can use PIMIM to jointly analyze a number of different conditions that allows to identify common and different like type condition-specific microRNA regulations. So last, I want to thank the sponsor for my travel work fellowship and also the funding agency for this research.
Yeah, thank you very much for the talk. It was very good in time, so we have time for questions. So, please.
I have a question about the basic biological idea behind your approach.
Yeah.
So your assumption is that if two proteins are interacting, they're likely to be co-regulated by the same microRNAs. Is
Yes.
that right?
Right.
How often is it the case that proteins that interact aren't co-regulated by the same microRNAs? And how often is it that proteins are co-regulated by the same microRNAs but aren't interacting?
Well, so, I mean, we can look at that from the results of the model. I would say, so in the paper we have a comparison that we compare the model with using the interaction data and without using the interaction data and compare what the result looks like, what's the benefits of using interaction data. And we found that I think the F1 score was significantly improved. I think it's going, the one I show you, I think 0.37. I think without using the protein interaction, it would be like 0.25 or something. So, and then the method that we compared against also use protein interaction data. So I think it's like significant. To know exactly the percentage, I cannot think off my head, but I think it's quite a high percentage.
You included all the predicted target relationships in your work.
Right.
And I wonder your module approach have the power of removing the false positive predictions in the results. So what would be the portion of your relationships validated and predicted in your resulting modules?
I see. So we try to, so far those like, so in terms of the interactions, we try to see if the one that we predicted are already validated biologically, like experimentally. But unfortunately, the database that available is so small, like, so it's like, yeah, it's so small that we get like, we're doing better than just like, just the second method that we compared, the method we compared to, but it's not like, it's like 0.005% or something. So I think it's, the next step we would, yeah, we try to validate some of it, but so far it's just, this is mainly like computational solutions. So yeah, so I don't have a good answer for you.
Some more questions? So I do not see any more questions, so thank you again.
All right, thanks.